Very good morning, all of you. Thank you, Dr. Christie, for inviting all of us or making us all come together to discuss on something which is more relevant on this very important day. The topic which probably I will be dealing with will be more to do with ectasia in general, with mostly all available literature, lot of work obviously is done on keratoconus and on PLE. So this would be sort of very generalized, but probably will be interesting. Let's dive into the basics. So somewhere in 2015, there was a global consensus that let's all group the whole ectatic corneal disorders together. So mainly the keratoconus, PMD, globus, and iatrogenic progressive ectatic disorders. When we look at PMD and keratoglobus, these are still in the rare, uncommon to rare category. So the available literature on the reasons and why, how will still be questionable, but most of it, as I said, is on KC and this one. All we do is basically when somebody comes with a very prominent ectasia, we say that there is ectasia. But behind this, there will be still those corneas which appear as normal, which tend to have or which can be labeled as ectatic disease based on some investigations. But what and how does this start? So let's look at the prevalence of probably all the ectatic disorders, mainly the KC and this one. An interesting meta-analysis which was published very recently looked into 7 million patients from 15 different countries, and they did suggest that there are some highly prevalent places and not so prevalent places. And some of this we found ourselves again not wanting to be there, but being there. India, Middle East, and Maori populations have a higher prevalence of keratoconus. If you look at the whole list of countries, somehow it appears that those countries which have more sunshine are more definitely having the prevalence more commonly. But does it mean that those in the less sunshine areas have? No. Interestingly, that's where the genetics take over. UK Asians have more commonly prevalent disease than the regular general population there. Again, it is the Indians, the Pakistanis, Bangladeshis who have more common prevalent disease, which is almost 4 times higher than the normal prevalence of that population at that particular place. So that's about keratoconus. What about ectasia in general? That is the refractive surgery ectasia. How do we know how many are there? Based on only published literature, it is not possible to even the numbers which somebody talks as the incidence really does not make sense there, because most of it can or will generally be underreported. Based only on the published literature, available literature, on an average internationally, nearly almost 1 million surgeries are done. 1 million refractive surgeries are done. This is an average. And out of which 1/9 will only be the surface procedure. Rest of it will be either LASIK. A few will definitely be, a few thousands to a lakh would definitely be smile. This keeps changing year on year. Okay. Obviously, the reason is it depends on when the whole thing started. Till almost only a decade back did US get this approval for doing smile surgeries. So the data available from there would still be limited. And the whole smile surgery only started 15, 20 years back, not even 20 years back. So it would be limited there. So when you extrapolate and try to work on numbers and some magic, you can arrive at some numbers. The numbers definitely say that PRK and smile have lower incidences compared to the regular LASIK. Again, that data can be challenged because over a period of time, in the last 1 decade itself, the overall development of science as such, in terms of screening technologies available, have tremendously increased, and hence the risk of PLE will be lesser than what is reported before. Let's look at it in two ways. One, let's look at the structure and then dive into what the chemical changes or immunological changes or histopathological changes. So in the histopathological change, when you look at a post-LASIK cornea, I did put up this instead of the regular keratoconus because I obviously did not get a good picture of keratoconus, but interestingly, everything will almost be same. So if you look at that, there will always be an epithelial hypoplasia above an ectatic area. The peripheral epithelia, where it is non-ectatic, will tend to be more hyperplastic. There would be some breaks in the membrane, but this breaks in the membrane is probably related to the keratome or this one. And interestingly, there will be this artificial artifactus lamellar cleft, which will be usually larger than the normal. That was about LASIK. About PRK, it's almost same, that the epithelial hyperplasia and this one would be same. Only thing is that the Bowman's would generally be absent, obviously be absent. And again, the clefts would be more than the normal. If you look at post-LASIK ectasia, LASIK after PRK, you will see that the LASIK intersurface wound, there would be some changes. Basically, it will be scarred over that area. So here it is in one picture talking about epithelial hypoplasia at the ectatic area, hyperplasia at the non-ectatic region, and a Bowman's break, which possibly can be seen as an arrowhead here in the. So this is again the same sort of a picture. Nothing much except that this one. So when you look at it, interestingly, there are all the lamellae are intact. The collagen probably at those areas would change in dimension, but the spacing is a problem. In this era, as a composite structure, a term used is there is an interlamellar fracture which happens. There is no breakage of any collagen. There is everything is intact except that they are more thin in the ectatic area compared to the non-ectatic. But because of this term, because there is thinning, there is the idea given is there is an interlamellar fracture, hence those areas are weak. When you look at the keratoconus, it's almost the same sort of physiological picture. There is, again, at the apex thinnest part, epithelia will initially be thinned out. As the disease progresses, the whole area can get thinned out. As the disease progression starts, the Bowman membranes becomes broken. So why is this happening? The next question is, we have seen some pictures and we have seen, but why is it happening? The obvious possible etiologies are based on some ocular and systemic factors. The ocular allergy, inflammatory factors, hormonal factors, cellular homeostasis factors, and some essential or trace elements which do play a role in the etiological factors. Most of us obviously now definitely understand that eye rubbing is something which is very, very important. Gatnell did establish that. In fact, even somebody who's dominant right-handed or left-handed have a more asymmetrical disease, the disease manifestation more on one particular side. So why does it occur? Is it because of a genetic predisposition? It was difficult to establish. The only way it was established initially was basically looking at a family history. Somebody has it, does anybody else in the family have it? It ranges based on publications. Our publications definitely have a wide variation depending on who is presenting, which country somebody is documenting. It is as low as 5% to as high as 1 in 4 will 25%. But most of it appears to be sort of an autosomal dominant trait, but with an incomplete penetrance. This is the standard established way of looking at things. But how is it causing is still a question mark, because there were some genes identified, but none of these genes have been able to establish that definite cause and association in this one. So the candidate genes which were identified and studied was something called as VX1 and superoxide dismutase 1. But the newer studies have sort of dismissed this idea. So the genetics still remains a poorly understood concept in keratoconus. When it comes to genetic syndromes, the challenge there is one, the disease itself is, the syndromes are itself pretty rare. And to say that it is a cause or an effect is always going to be a challenge. UV light, because we say it is common in more sunlight, we do understand that it is important. But again, on the contrary, without UV light exposure, there is no cross-linking. So this is an interesting one. Occurs in more sunny areas where you say UV light is more, but then you use UV light to treat or to increase cross-linking. So when you talk about allergy, both the ocular and systemic allergy, eye rubbing, as we said, is obviously a definitely established factor. What happens here is the immunoglobulins, specifically IgE levels are high, histamines are high. There's another important entity called as matrix metalloproteinases. Most of us would have understood that it's sort of a proteolytic enzyme. This are many in numbers. More importantly, the 2 and 9, MMP2 and MMP9 are elevated in the cornea. The problem here is there is a proteolytic and an anti-proteolytic activity which keeps homeostasis. Whenever the homeostasis there's an imbalance in the homeostasis, the MMP9 triggers up and that starts causing proteolysis or destroying this one. To balance it, there is something called as TIMP, tissue inhibitors of matrix metalloproteinases. They are somehow less in keratoconic areas or eyes. So where do we get it? It's available in the tissue and also possible to get in the teophyllum. Does it have a clinical implication, or can it be used for identifying patients? It can be used for identifying patient. Is it a sine qua non that it will be the only reason? No, it is not the only reason. So it is just a test which is available now. Dr. Rohit Shetty and team have even come out with a pocket sort of a identifier where you will be able to measure it. How do we treat it? We don't know, but we know it can be raised even in dry eyes. The next one is lysyl oxidase. This is an enzyme which is very crucial for maintaining the collagen cross-links. So this converts lysine residues in the collagen to aldehydes. And because of this conversion, the cross-links becomes more stable. So whenever the lysyl oxidase activity decreases, there is increased risk of formation of keratoconus. So that is basically, there is weakness and hence keratoconus. So you look at the inflammatory markers, a lot of inflammatory markers have been studied. So the classical ones like the interleukin 1, alpha, beta, they do promote collagen degradation, then the extracellular matrix remodeling, and definitely contributes to corneal thickening in general. But how much of this each one interplay is a complex entity. So the interleukins also raise in other forms of inflammation. So allergies, when we said that eye rubbing is almost sine qua non with keratoconus, in most of the allergies, some of these inflammatory markers also raise, the interleukins also raise. The IL-4 and IL-5 are definitely seen in allergic inflammation. The IL-13 also is seen in an allergic response. So there are some pro-inflammatory cytokine, that is the TNF-alpha, which activates the MMPs, which thus also breaks down and weakens the cornea. So coming to the hormonal factors, recently there is an understanding that there are receptors on the corneal epithelium itself, both the sex hormones, whether it is the estrogen or the progesterone, and androgen, there are specific receptors on this one. So it is seen that there's a decreased androgen precursor and increased estrogen hormone levels, which together are detected with an altered expression of prolactin-induced protein. And this is used as a marker for KC progression in one of the studies. So hormones do play a role, and we do understand that during pregnancy, the ectasia worsens. And those who are predisposed by genetic, this one, in them, the ectasia manifests. Some studies, there's an interesting study in some Germany, where it said that thyroid dysfunction is almost, is commonly seen in people with keratoconus. How is directly related? Not yet established. Trace elements. Most of these trace elements do have a definite role to play in when you look at individually on the role of collagen synthesis. So the copper is an important cofactor for LOX. So if there is copper deficiency, LOX function depletes and the collagen cross-linking reduces. Zinc is involved in various enzymatic processes, basically related to production, collagen production and repair. Selenium as an antioxidant in general has its role in protecting tissue. Calcium generally is important for the overall tissue integrity, including that in the cornea. Manganese is important for collagen synthesis, and magnesium for stability. All these, just like the trace elements, do have a trace activity which is difficult to decipher, but are all important. So do we need to do all those tests and establish? No, it may not be relevant as of now. So we sort of try to run and understand the etiological factors. So there are some numbers. So what is that pathway? How does this convert? So basically, there has to be a genetic predisposition. If there is, with or without, if there is a prediction, there is reduced collagen expression in the extracellular matrix. Then there is alteration in the proteoglycans and the glycosamine glycate. Then there is reduced endogenous cross-linking and enzymatic activity. All this also leading to an increased proteolytic enzyme. So the extracellular matrix play an important role in maintaining the homeostasis. Oxidative stress, something which is just like our stress. We know that it is there, but we don't know how it is this one. So there is this reactive oxygen species which increases whenever there is oxidative stress. It is balanced by the superoxide dismutase, which is the basic an antioxidant. So that's important. And there are some oxidative DNA and protein damages, which are also seen because of all the inflammatory markers which were there. HSP, the heat shock protein 27, increases in a response to oxidative stress. Basically, again, it is one of those antioxidant substance which is there, which is working against the ROS, the reactive oxygen species. We talked about cytokines. Cytokines play a very important both in the collagen and the extracellular matrix. So all this finally leads to some form of a biomechanical change, which leads to thinning of collagen. The spacing between the collagens in the in the ectatic area is sort of widened or irregular. That leads to some morphological changes which we pick up at our diagnostic equipment. So this manifestation is finally what we see as ectasia. So you have a set of etiological factors which is there in everywhere, but unless you have a genetic predisposition, or because either intrinsically because it is there, or extrinsically, sorry, when you do a surgery, when you ablate the cornea, you make the cornea more thinner, or you start rubbing more, this pathophysiology will finally lead on to a manifestation which we call it as ectasia. Thank you.